Hello everyone. So today we'll be doing about the chapter molecular basis of inheritance. Okay. So the first thing in this chapter is the DNA. Okay. So the full form of DNA is deoxyribonucleic acid. Okay. So what are DNA? See, DNA are a long polymer. Okay. It's a long polymer of nucleotides. Now see, the long polymer means more than one. Poly means more than one. Okay, so it's a polymer of nucleotides. That means DNA is made up of nucleotides. Okay, nucleotides. So it's a polymer, that means long chain of nucleotides. That means more than one nucleotides. Okay, it goes on. Okay, it goes on. Okay, so DNA is made up of more than one nucleotide 1, 2, 3, 4, 5, and so on. Okay, so we can say that the length of the DNA, okay, any DNA of human beings, DNA that is present in the cell of any living organism, okay, we can define that the length of the DNA is defined by the number of nucleotides present in it. Okay, so how many nucleotides present in a DNA will determine the length of DNA, because DNA are the polymer of nucleotides. Polymer, that means it's a long chain of nucleotides. All right, this is just the rough diagram, okay, in order to make you understand. Only nucleotides are not like this, not this square shape. Okay, this is just to make you understand. Okay, so it's a, now you have understood it's a long polymer of nucleotides. Now while talking about these nucleotides, okay, one single nucleotides, see, one nucleotide, we can say that nucleotide are the unit of DNA. Okay, nucleotides are the units of DNA. So when we talk about one single nucleotide is made up of three components. Okay, one nucleotide is made up of three components. And what are these components? Number one is nitrogenous base. Okay, number two is deoxyribose sugar. Sometimes it is also called pentose sugar. Okay, and the third one is your phosphate group. All right, okay, so see, DNA is made up of more than one nucleotides. That means polymer of long chain, or polymer more than one monomer. Okay, more than one unit, more than one nucleotide. So long polymer of nucleotides. And one nucleotides further, they are composed of nitrogenous base, deoxyribose sugar, and phosphate group. Okay, while talking about this nitrogenous base only, and further this nitrogenous base, there are also four different types. Okay, there are four different types of nitrogenous base: adenine, guanine, thymine, and cytosine. Okay, so there are four types of nitrogenous base. Now you can see in your book also, NCERT book, it is given, clearly given. Okay, Now see, if this, I'm drawing only this one particular nucleotide, okay? If this is a pentose sugar, this is pentose sugar, then, okay, this is 5 dash, okay, 5 dash OH group. This is 5 dash OH group here, okay? And here it is 3 dash OH group of pentose sugar. All right? Now see how the different nitrogenous base, deoxyribose sugar and phosphate group, they are placed in a one nucleotide. See, if this is a pentose sugar, okay, this is only the pentose sugar, that is deoxyribose sugar, then the 5 dash OH group here, okay, the 5 dash OH group of this ribose sugar is attached to a phosphate group. Okay, this phosphate group is linked to this ribose sugar, where 5 dash OH group of this ribose sugar. All right, and here we have, okay, and here we have nitrogenous base. Now nitrogenous base can be not all nitrogenous base will be in one nucleotide. One nucleotide contain only one nitrogenous base, and that nitrogenous base can be either adenine, guanine, thymine or cytosine, sorry. Okay, so suppose this has guanine. Okay, so now this complete structure is our one nucleotide. Similarly, 1, 2, 3, 4, many nucleotides are linked together in order to form a long chain of polynucleotides. More than one nucleotides, polynucleotides. So DNA are a polynucleotides chain also. Alright. So DNA are polynucleotide chain. And see, so this way it continues, okay? There are so many nucleotides. I have drawn only one single. This is single nucleotides. This is ribose sugar, deoxyribose sugar. In the 5 dash OH group of this ribose sugar, phosphorus group or the phosphate group is attached, okay? And guanine, adenine, thymine, cytosine, any of these, okay, nitrogenous base are also attached to the deoxyribose sugar. So this way one nucleotide is formed, okay, and many nucleotides, they come together, they join together, okay, and forms a polynucleotide chain. Now how it is done, we'll come to know later on, okay? All right, so this is all about the introduction. Now very important, another thing is this DNA, okay? When we talk about DNA is not only one polynucleotide chain, okay, not one polynucleotide chain. If this is nucleotides, okay, see, DNA does not have only single strand. DNA also have another strand, okay, because DNA is double stranded. Strands are nothing but this DNA, okay, chain of nucleotides, polynucleotides chain, okay. So see, DNA is made up of two polynucleotide chains, not only one, okay, and these chains are not straight, okay. I have drawn like this way just in order to make you all understand, okay, but these two polynucleotide chains, they are helically coiled structure, okay, they are helically coiled structure, okay, they are not straight like this, they are not parallel like this, sorry, parallel, yeah, they are parallel, but they are helically coiled structure, okay. That means DNA is double stranded. That means it is made up of two polynucleotide chain, not only single polynucleotide chain. Okay, so this is just the introduction. Now so we have also come to know that the length of a DNA, okay, length of a DNA in any cell of any living organism, it is determined by the number of nucleotides present in its DNA. Okay, the more nucleotides present in the DNA, the length of the DNA will be longer. So okay, so this is all about the introduction. So okay, now coming to the structure of polynucleotide chain of DNA. Okay, first thing is nucleotide is made up of 3 component: nitrogenous base, deoxyribose sugar, phosphate group. Then these nitrogenous bases are also 4 types in DNA. Okay, adenine, guanine, thymine and cytosine in DNA. Okay, but in RNA, in place of thymine, RNA have uracil. Okay, RNA have adenine, guanine, uracil and cytosine, but DNA have adenine, guanine, thymine and cytosine. Okay, now this nitrogenous base, okay, when we talk about this nitrogenous base, there are four nitrogenous base. Okay, but when we talk about types, okay, these 4 nitrogenous base are grouped into 2 types. Okay, these are grouped into 2 types, nitrogenous base. Okay, nitrogenous base, 2 types, purine, okay, and pyrimidine. Purine and pyrimidine. Okay, purines are adenine and guanine. This adenine and guanine, they are called purine based nitrogenous base. Okay, purine nitrogenous base. Pyrimidine are thymine and cytosine in case of DNA, but in case of RNA, in place of thymine we have uracil, so pyrimidine will be uracil and cytosine. Okay, all right. Now how the different nucleotides are linked, we'll be doing that one. Okay, now it is given in your book also. Okay, I've told you, if this is sugar, okay, deoxyribose sugar, if this is the nitrogenous base, and if this is phosphate group. Okay, now see, very important, we are talking about linkage, how different components are linked together to form a nucleotide, and how different nucleotides come together and linked and joined together to form polynucleotide chain. Okay, see, nitrogenous base, any, okay, nitrogenous base, it can be adenine, guanine, thymine or cytosine. These are linked with the deoxyribose sugar, okay, through this linkage. Okay, this linkage is called an glycosidic linkage. Okay, nitrogenous base is linked to deoxyribose sugar through an glycosidic linkage. Okay, now when these nitrogenous base, they get linked with deoxyribose sugar, then this structure, deoxyribose sugar plus nitrogenous base together, this structure, when they link together, Okay, when phosphorus, this phosphate group is not linked yet, but only these two are linked first. Okay, this linkage are called nucleoside. Okay, these two, when they link together, nitrogenous base and sugar, they are completely not a nucleotide. Okay, because they are still not yet linked with phosphate group. Now these two only, when they are linked, these two only, this structure are called nucleoside. Nucleoside, not tide. Okay, nucleoside, SID. Okay, so now this nucleoside, okay, it can be four types. Why it can be four types? Because our nitrogenous base can be any. It can be adenine or thymine or guanine or cytosine. Okay, so depending on which nitrogenous base is linked to the pentose sugar, depending on which nitrogenous base is linked to the pentose sugar, nucleoside can be four types. Okay, yeah, it can be deoxyadenosine or deoxyguanosine or deoxy, this is one single word. Okay, cytidine or deoxythymidine. So these are four names. Okay, depending on the type of nitrogenous base which is linked to the pentose sugar. Okay, yeah, so there are four different types of nucleoside. Now how nucleotides are formed? Okay, when this nucleoside, okay, when this structure now linked with the phosphorus, where 5-OH group of this pentose sugar. Okay, when the phosphorus is linked to the 5-OH group of this ribose sugar, then at last when deoxyribose sugar, okay, plus nitrogenous base plus phosphate group, they are linked. Okay, all the three components are linked together, then only it will form nucleotide. All right, so they are linked at 5-OH group of this ribose sugar. So nucleotide are also of four types. Okay, now, and what is the name of this linkage when phosphorus is linked to pentose sugar? This linkage is called, okay, this linkage is called phosphoester linkage. Okay, when phosphorus gets linked with ribose sugar at 5-OH group of ribose sugar through phosphoester linkage, okay, then this whole structure together, it is called nucleotide. Similarly, so many, this is only one nucleotide. Okay, so many nucleotides, they come together, they link together and form a chain of nucleotides. All right, so, and not only one chain, DNA is made up of two chains. Okay, now, okay, I'll just show you the book. I think it's given in the book also. Okay, just see here. When you see your book, okay, when you see your book, here it is clearly given. Okay, phosphate group, this is ribose sugar, and adenine, this is one nucleotide, two nucleotide, 3, 4, and so on. Okay, throughout the length of DNA. Okay, so this linkage, when phosphorus is linked with ribose sugar, this linkage is called phosphoester linkage. Okay, when nitrogenous base is linked with ribose sugar, this linkage is called, yes, N-glycosidic linkage. Okay, now there is another type of linkage. Now, which type of linkage? How two nucleotides are joined together, more than one nucleotides joined together or gets linked together, it is through 3'5' phosphodiester linkage. That means, now if you see this diagram, okay, second nucleotide, you know this is 5', okay, this is 5' OH group where phosphorus is linked, isn't it? 5' OH group of ribose sugar. Now this phosphorus linked with 3' OH group of the another nucleotide, another ribose sugar of another nucleotide. Okay, similarly, now this is 3' OH, and here in this it is 5' OH. So that's why the name of the link is called 3'5' phosphodiester. Diester. Okay, when phosphorus is linked with the 5' OH group of this nucleoside, at first it is linked by the linkage called phosphoester linkage. Okay, when 2, 3, 4, so many nucleotides come together to form a link, then the linkage is called phospho 3'5' OH, okay, of the adjacent ribose sugar, okay, adjacent ribose sugar of two nucleotides. That's why the linkage is called 3'5' phosphodiester linkage. Okay, and this nitrogenous base can be any, okay, A, T, G, C, C, any. Okay, all right, so yeah, now coming to the salient feature. Okay, see, DNA is also called, this DNA is also called, I've already told you, double helix structure. Okay, double helix means DNA is not only made up of one chain of polynucleotides, okay, but it is made up of two chains of polynucleotide which is helically coiled. That's why helix, helically coiled. And see, the double helix model of DNA was proposed by the scientist Watson and Crick. Okay, Watson and Crick. It was given by James Watson and Francis Crick. Okay, full form you can get in the book. Sorry, not full form, full name of this scientist, two scientists. Okay, what they have told is that double helix model. Okay, now when this okay, when I take a segment of DNA, if I draw like this only, it is not like this. Please remember that it is always helically coiled, just for your knowledge, in order to make you understand. So there are so many nucleotides, 1, 2, 3, 4, 5. Similarly, 1, 2, 3, 4, 5. Okay, two strands are there. Now this is one nucleotide, this is another. Similarly, 1, 2, 3, 4, 5. Okay, now one nucleotide consists of phosphate, sugar, and one nitrogenous base. If suppose it is adenine, okay, so nitrogenous base, okay, nitrogenous base. Similarly here also nitrogenous base, okay, and here they have phosphorus, isn't it? Phosphate group, okay, phosphate group. Okay, so these are two strands of DNA. So nitrogenous base can be any A, suppose T, if this is G, this is C, okay. Now very important, when these two strands of DNA come together, okay, see, adenine always pair with, if there is adenine, then there must be thymine here. Adenine always pairs with thymine, okay. If there is A, that means here it should be T. Adenine always pairs with thymine. If there is T, there must be A, okay. Complementary base pairs, they are called complementary base pairs. If there is G, there must be C here, okay. So here it is C, so there must be G out here. So guanine is always complementary to cytosine. So these pairing, when adenine pairs with thymine, okay, adenine pairs with thymine, guanine pairs with cytosine, or vice versa, okay, then it is because they are complementary to each other. Adenine is complementary to thymine, guanine is complementary to cytosine, okay. Now here you can see when adenine pairs with thymine, I have drawn two lines, isn't it? So these denotes adenine pairs with thymine, adenine, okay, thymine 2, and guanine pairs with cytosine 3, and here also 3. This 2 and 3, this denotes hydrogen bond. Adenine pairs with thymine by 2 hydrogen bond, always remember, and guanine pairs with cytosine by 3 hydrogen bond, okay. And this adenine, guanine, these things, okay, they comes inside pairing, okay, pairing, they come inside, and phosphate group, they lies outside towards the back, okay, towards the back, like this. Phosphate group towards the back, like this, and this nitrogenous base, they form, this pairing takes place inwards, okay, inside. So that's why we can say that ribose sugar and phosphate, they forms the backbone of DNA. They form the main structure of DNA, okay, backbone of DNA. This ribose sugar, deoxyribose sugar, and phosphate group, they forms the backbone, and the nitrogenous bases, they lies inwards, inside, they lies inside, okay. All right, now another thing is that see, so again I'll show you the book. Here it is given, clearly given, okay. So in this diagram, I hope it's visible for you all, okay, in this diagram you can see here, this is one nucleotide, okay, one nucleotide, another, all right. This is just the segment of DNA, small segment of DNA, okay. Similarly here also 1, 2, 3 and 4. Now you can see here in one nucleotide there is ribose sugar, nitrogenous base and phosphorus. Now when you look into this structure or this diagram, you can see here adenine is pairing with thymine. Always remember, in DNA adenine always pairs with thymine by 2 hydrogen bond. Okay, bond is also given, 2 hydrogen bond. Okay, thymine pairs with adenine by 2 hydrogen bond. So this is one polynucleotide chain, this is another polynucleotide chain. Okay, you're also given one polynucleotide chain, another polynucleotide chain. That's why DNA is double stranded. One strand, two strand, double stranded. Okay. Okay. Guanine is pairing with cytosine by 3 hydrogen bond. Okay. So now I hope this picture is clear for you all. See, yellow color denotes adenine, green color is denoting thymine here. Okay, just to make you understand, it is not that DNA is colorful and adenine is yellow in color. It is not like that. Okay, just in order to make you all understand, it is given like this in the book. So when you see this diagram, green, that means thymine. Thymine is pairing with adenine, because I have already told you these two are complementary bases. Okay, thymine and adenine, adenine are complementary bases. Adenine cannot form any link or they cannot pair up with guanine and cytosine. Okay, thymine cannot pair up with guanine and cytosine. Similarly, cytosine cannot pair up with thymine and adenine, and guanine cannot pair up with adenine and thymine, because they are not complementary. Complementary are adenine and thymine, okay, and guanine and cytosine. Okay, so if there is cytosine, in the another strand there must be guanine in order to pair. Okay, so it is structured like that only. Okay, so you can see here sugar, so this forms the backbone, this strand. Okay, so this strand, they have sugar and phosphorus. So sugar and phosphorus, they form sugar phosphate backbone. They give the structure to the DNA, and this inward towards the inside, okay, inwards lies the base pairs, complementary base pairs. Okay, so this is all about the structure of DNA. Please do read your book so that you will get more clarity. Okay, all right, so in the next video I'll be doing about the salient feature of double helix structure of DNA. Okay, thank you.